Dr. Mamata M. Pillai, she is now working as a project research scientist, Department of Bioscience and Bioengineering at IIT Bombay, India. She has completed her PhD at PSG Institute of Advanced Studies, Bharatiya University, Coimbatore, at 2017 in the field of nanobiotechnology. After then, she has joined as a postdoctoral researcher in the same institution. Then did her postdoc for 1 year, and then she shifted to IIT Bombay during 2019 to 2022. She has completed another postdoctoral degree in the same area of research. Since April 22, she is working as a project research scientist. She has a vast research experience of more than 10 years in the areas of immunotherapy, tissue engineering, regenerative medicine, drug delivery, nanofibrous transparent face mask, 3D lithography, and she has received a Marie Young Scientist Award in the consecutive years 2020 and 2021, and Young Researcher Award. And also she is one of the NIDI EAR Fellow, that is Entrepreneurs in Residence, awarded by Department of Science and Technology, India. And she also got Rising Star Award, apart from many poster and eventual awards. To her name, she has filed 4 patents and published more than 36 international research articles. And she has written 2 books and 3 book chapters. So with this brief note, now I ask the resource person to start the presentation. Thank you.
Thank you. Thank you so much. A very good evening to all. Thank you, Dr. A. Subramanian, Principal of the college, and Dr. Silambarsan for having me here. And thank you so much for the nice introduction which you have given. So allow me to share the screen first. Whether the screen is visible to all?
Yes, ma'am, it's visible.
Okay, I'm putting in slideshow. Okay, then I think after this lunchtime, it will be very hectic to hear any lecture, I feel, for students. So anyway, I will try to, give some basic idea of tissue engineering for biomedical applications. So the word tissue engineering is a little bit fascinating, right? Why? Because it is not that old scientific field, but the emerging technologies which has come up recently are very fascinating. Like you can even print an organ, a complete organ like a heart or kidney or something like that, that can be implanted. So it looks like very, kind of promising results are coming, but nothing is coming to the industrial market, or you cannot buy anything and go and buy directly. Why? Why? I will let you know during my presentations also. So coming to the word tissue engineering. So if you split the word into 2, tissue is engineered. Tissue means our cells, which is made up by the tissue, and then organ. This can be engineered somehow. How it is like? Langer and Vakanti, 2 scientists, they have defined this term. Tissue engineering is like an interdisciplinary field which applies the principle of engineering and life sciences toward the development of biological substitutes that can restore, maintain, or improve tissue function. So how it is restored or maintain the tissue functions are like, I will just give my pointer option, laser pointer. So here you can see an image, right? So how the tissues are maintained or restored or improvised is like, we can use different aspects. Like material is one part. Some sort of biomaterial is required for engineering the tissue. And of course, as tissue is there, this means it has something that is viable or that is living in the sense. Cells. Those are the cells which make up multiple cells combines together to form a tissue. and tissue will form as an organ system, right? So the basic unit of life, cells are required to regenerate any particular organ. And then we require some physiological active substances like some growth factors or signaling molecules, which are required for the cells to proliferate and to join together and to form a tissue. That's how the function of the tissue is restored and maintained, or we can even use it as a substitute for many organs or tissues, is what the idea of tissue engineering in a broader perspective. So how we can make such kind of implants or organs is what I'm going to explain. So there are many tissues named or listed here, like nerves, cartilage, gums, bone, blood vessels, bladder, eyes, skin, tendons, muscle, pancreas, cardiac muscle, liver, everything we can make except our brain. So brain you cannot 3D print, even that is the highest or advanced version of tissue engineering we can say. Our brain cannot be made synthetically or naturally by using any such kind of modification or improvement while we are doing or developing such kind of technologies. So there are different organs which are not able to, mimic, or which we are not able to develop. So which are doable, I will be explaining in my coming slides. So this tissue engineering strategy or tissue engineering triad is known as or comprises of these three components, which I have already mentioned. There should be a biomaterial source, there should be cells, and then signaling molecules, where we can do the engineering aspect, and then which will be making, which can be developed into something that can be implanted on top of any damaged tissue. For example, I have shown here a wound. So after tissue engineering, you will get a scaffold, we can say, term it as a scaffold, and this can be used for tissue engineering aspects. So these terms, what all are there, one by one we can go through. So the first thing is the cells, right? So I am going to some basic biology. You can, recollect your biology of like maybe 6th or 7th standard, where structure of human cell and plant cell, which we have studied, right? So the human cell, a little bit complicated. It has a lot of organelles. You can see here mitochondrial, lysosome, nucleus, which has its own genetic makeup like RNA and DNA and everything. And then endoplasmic reticulum, microtubules, and different sorts of organelles, which has sophisticated function. You can see it here, right? So these cells will not grow or proliferate like this. So the cells require some specific environment to grow and proliferate, like within our body. Like each cell, single cell, can make up into a tissue, and this multiple tissues can join together to form an organ, and this forms as an organ system, right? So this, in our body we have blood circulation, we have other waste management system, where through kidneys and everything, the waste and everything will go, and liver will help in metabolism. And there are, even each cell has its own metabolic capacity, like it produces ATP for its energy and everything. So everything bringing to an in vitro system or in laboratory level or in a test tube is very difficult. We have to give each and every aspect to bring into a lab scale or an in vitro scale, we can say. So in vitro is nothing but, doing it in a culture plate or do it in a test tube. You have maybe heard about test tube baby. You have maybe, that is a really fancy word, test tube baby. The baby is not born on a test tube, but the cells are, processed or programmed in a kind of a culture vessel. That's why it is known as in vitro. So what all type of cells are there basically, which we can use for tissue engineering are, one second, I'm not able to change my slide because of this pointer. Automatic. Okay. Okay. Sorry. So there are different types of cells. Mainly, for tissue engineering, widely we can use is stem cells. Maybe you have heard about stem cell therapy, maybe like embryonic stem cells and all. Now storing, they are, just after birth of the baby, the embryonic stem cells can be stored in many hospitals, where, because it can be used for future for different types of treatments. So the stem cells are widely being used. There are different types of stem cells. Why stem cell is because one stem cells can be regenerated into or differentiated into different types of tissue cells. For example, one stem cell can be differentiated into bone cells, not stem cells, sorry, bone cells, or it can be differentiated into a skin cell. It can be differentiated into corneal cell, which is our corneal, which is seen in our eyes. So there are different potency, that is known as potency of cell, that is pluripotency. So pluripotency means one cell can divide it into different types of cells. So that is known as pluripotency or multipotent stem cells. Totipotent stem cells are like, it can differentiate into a complete organ. That is embryonic stem cells. You see that the ovum, the one single cell can come into as an organism. Those are known as totally it is potent to become an organism. That is known as totipotent. So there are different types. Many cells has, its own advantages and its own disadvantages also. So like if we say like there are different types of stem cells, it will be difficult for you to remember each and every word. So I'm not going in much into detail about its scientific aspect. But if you see the tissue derived stem cells, why the main disadvantages are like that it's difficult in isolation from the tissues, because your tissue is made up of different types of cells. So if you want to pick only one cell out of that different population of cells, it will be a tedious process. So that's why mostly embryonic stem cells or induced pluripotent stem cells are preferred for doing tissue engineering. So coming to the in vitro cell culture, as I told you, it requires a very sophisticated sterile facility. That means sterile in the sense none of the organisms should come into, it will contaminate your cell culture, or it is like a class 2 level biosafety is required, so that we also should not get any infections, or from our side the contamination should not go into or go onto this kind of cell culture which you are doing. So first step is like for in vitro cell culture, we have to isolate the cells. As I mentioned, it can be from a bone marrow if it is stem cell, or you can purchase the cell lines which is available in ATCC, that is American Tissue Culture Center is there, or you can get it from any other cell source. There are a lot of cell source available. We can purchase directly from them to unnecessarily do a laborious isolation process, like you have to treat the cells with enzymes, it has to be centrifuged, it has to be maybe if it is like a co-culture, you can see it's like two different cells are there, then you have to pick only one colony out of it. Then it is again you can use flow cytometry like FACS or something to sort the cells out, to purify the cells further, to get into one single phenotype of cells. So this is a very tedious task. You have to always maintain the purity, its phenotype in the sense morphology of the cells, its metabolic activity of cells, like whether it is secreting a particular type of metabolites, like its extracellular matrix, whether it is functional. Then only we will confirm that the cells are functional, the cells are in this desired state of replication or multiplication, we can say that. So there are different sterile culture weighers are used because, as I mentioned, it is in vitro. So there should be something like a test tube, like a, we are using, this is a 24 well plate, this is cell culture flask. There are different types, different sizes of flask, which require, if you require very small volume, we can use 24 well plate. If it is larger volume, we can use 100 mm dishes or tea flask and all. So this is just an image of biosafety cabinet where we can do the cell culture. And other thing is like we need cell culture medium. As I said, in our body, the cell, while the cells are growing, they will be getting the nutrients and everything through our blood circulation, right? So here, once we are isolating it, it can die immediately or within few hours. So we have to provide certain conditions, certain temperature, certain nutrients for these cells to make these cells, or to maintain these cells in its phenotype and functionality. So such kind of cell culture media are available, for example, RTMI media, DMEM media, along with we have to use some fetal bovine serum. So The bovine serum is an additional nutrient which is required for the maintenance of the cells. And depending upon the cell type, depending upon the differentiation factors, there are different growth factors or factors which you have to use to maintain or differentiate the cells. So this is how the cells will look like. These are the single cells. As you can see, there are some type of morphology you can see. We can say it as an elongated morphology of cells you can see here. So this type of, this we require a phase contrast microscope, which is another sophisticated equipment which we will be able to view or visualize the cells under the microscope. So this is how the in vitro cell culture happens. So once we know that which cell has to be used for tissue engineering purpose, those cells can be isolated. It can be grown in vitro culture, and once a sufficient number of cells are ready, we can go for the next set of experiment. So here again, I'm coming to the same picture because just I have mentioned about the cells. Now we will go to the biomaterial based scaffolds. So let your cells get ready there. Now we will make the second part ready, that is the biomaterial part. So coming to the biomaterial part, so biomaterial design is also very critical. If you don't know what is the chemical make or the toxicity of the biomaterials, you will not be able to use it as a use it for making scaffolds. So it is very, very important to design your biomaterial according to your need. For example, that there are soft tissues and hard tissues, right? Like bone, if you see, you cannot crush it with your hands, but liver you can. Liver is very soft, you can crush it with your hand. So for the both application, you cannot use the same biomaterial. That is obvious. So we have to tweak the biomaterial properties in such a way that it can be used for both the applications. So there are different biomaterial sources like synthetic biomaterials, and also natural polymers are also available. For example, polycaprolactone, polylactic acid, polyvinyl alcohol, collagen, hyaluronic acid, chitosan, sulfibroin. You can, there are lot of which we cannot enlist in one single slide. So there are a lot of polymers are there in this, universe, where we cannot use everything, but we have to choose according to our, need. So that is very critical. So some key factors are there for the biomaterial. The source is very important. So if you're isolating some collagen from pig and trying to implant it in human, obviously there will be some immune reactions or rejection will happen, right? Because it is not of the same source. So the source of biomaterial is very important. Otherwise, you can choose a biomaterial source in such a way that there will not be any immune reaction will happen when the biomaterial is implanted. And mechanical property is very, very important. As I mentioned, as I given the example before, the bone and liver or skin, if you take the skin is highly elastic. Skin is highly elastic, but bone is what? Highly hard, hard material. So for different type of applications, you have to choose a single biomaterial or combination of biomaterials which can mimic the mechanical properties, biological properties of your desired tissue. Next is biocompatibility. Of course, you cannot use non-biocompatible materials. Biocompatible in the sense it should not give any toxicity. If you use any acid prepared or acid synthesized polymers and everything and trying to put into the body, obviously there will be some reaction because the pH balance is gone there. Immune cells is coming to attack your, biomaterial which is implanted, and it's going to be completely getting rejected. So that will exert to further complications which is not required, because we have list of polymers which we know that okay, this is biocompatible, this can be used for bone, and this is another polymer is biocompatible, this can be used for skin tissue engineering. So that's how you have to design your biomaterial in such a way it should comply the need of the desired tissue. Next is the bioactive factors which we can use. So there are different bioactive factors available, like which will enhance the bioactive factors, which will enhance few kind of properties such as it will enhance, it will act like a growth factor. For example, growth factor in the sense, uh, as I mentioned, in our body, there are a lot of blood vessels, right? So these blood vessels will only provide the nutrients to each and every part of organ, and finally it reaches to a single cell also. So here we are coming to an in vitro system where there are no blood vessels at all. So there is only some material and there are some cells. We have put it into a culture medium. The material, the cells should get some nutrients. So how it is possible? How we can make the blood circulation into this kind of materials which we are developing in vitro? So there we require some growth factors like vascular endothelial growth factors. The blood vessel cell is known as endothelial cells. So then we can use such kind of vascular endothelial growth factors, and we can culture this endothelial cells along with your desired tissue cells to get vascularity inside your scaffold. Such kind of growth factors are very, very important. You have to, it is very expensive, and you have to optimize the concentration in such a way that it will not get into some kind of mutation or cancer level, because over proliferation can happen. And some sort of chemokines can be used. Chemokines in the cells, chemical attractants. So where like you can make a scaffold acellular, that is without seeding any cells, you can implant it, but you have some chemokines. So you have some chemicals which attract cells. So in your scaffold, if you are adding some chemokines, the cells will, this chemokines will start releasing from it, and cells is going to come into the scaffold and sit. See, this is also very fancy, right? So it happens. So this chemokines and this kind of chemical attraction happens. That can also can be done so that you can avoid such laborious process of preparation of your cells and growing in vitro and everything. And some few drugs for therapeutics, like if you want to regenerate or heal wound, so such kind of wound healing purposes, you can add some drugs into that and directly give it. And then phytochemicals also you can use, like curcumin, honey. There are a lot of phytochemicals available which has, you know, anti-inflammatory, anti what, there are a lot of antibiotic, then any other, you know, enhanced cell proliferation activity, and all these things you can give. And there are different types of scaffold as I'm given here. There are hydrogels, nanofibers, there are decellularized scaffolds. The decellularized scaffold in the sense, for example, you have, if you're taking a pig or any tissue like kidney, you can remove the cells from, there are a lot of chemical process that can be remove the cells from that. So what you will get is kind of a kidney without cells. Now what you will do, you will process it, its antigenicity you will remove, and then you will use the same kidney for reseeding the cells. And then the functionalized kidney, once it is regenerated, that can be used as an implant. So these are decellularized scaffolds, which is a widely known practice for doing regeneration of tissues. Next is biofilms. As you can see, the artificial skin is like a film, a thin film which is not having that much thickness. The skin is having 4 to 5 mm thickness, right? So that much thickness, we don't need a hydrogel or something to regenerate the skin. So the film form is better. Then electrospun scaffold also, which will have very less thickness. It will be in few millimeters. So that also can be used for different applications for drug delivery or, you know, for wound healing and all. And you know, 3D printing, how the 3D printed scaffolds will give precise structure, dimension for the cell to proliferate and everything, or you can add cells also into that, and you can print it out and use it as an implant. And a hydrogel is something like which will have, it is hydro plus gel. Hydro means water plus gel. So it already has water inside that, and it is in a gel form, jelly like you know, the edible jellies which is coming, it will be in that consistency, so that the cells can grow inside. So it will have less pores, so it cannot be used for many applications, and it don't have such mechanical properties also. So that's why hydrogel can be used only for soft tissues like liver or something like that. So as I mentioned, polymers, the polymers, how I made, I told initially, like there will be monomers, and these monomers, through a polymerization chemical reaction, this polymer chain will be made. So each polymer will have particular molecular weight. So according to that molecular weight, only the polymer chain length can be determined. So like PVA, you will get a molecular weight of 3000, you will get a molecular weight of 5000, you will get a molecular weight 8000. So this also will matters, like each polymer has its own, depending upon the chain length, the molecular weight also varies. So within the same polymer, how you will choose different molecular weight. So that also is according to your application which is required. For example, if you need liver, so liver is very soft in nature, so you can go with a low molecular weight. So if you, the same polymer, if you want to make it in a higher thickness, like it should have a extensive elongation or something, you can go for a higher molecular weight. So these are all, some tips or something like for just one FYI I can give to you. So the polymers, as I mentioned, I have listed in the previous slide also. So there we should use inside our body for tissue engineering. The main aim is to use biodegradable polymers. That means it should degrade or it should dissociate after some time. So we have a scaffold made up of, I will tell you what is scaffold. There is a structure, support structure is there, which is this polymers, why we are using is to develop scaffolds, right? So this scaffold should degrade at a particular time by, like we have cells or something added, right? It will secrete its own extracellular matrix. It will form a organ. After that, why this biomaterial is there? It should not be there. It should go away. Then only the tissue can function properly. So this classification is known as biodegradable polymers. There are non-degradable polymers also, like plastic, that is a polyethylene or polyester kind of plastics are available, which we cannot use inside our body. Or there are metals which cannot be, which is generally used, but these are not biodegradable. They are permanent implants. So tissue engineering, we can make permanent implants, but it is, this tissue engineering main idea or focus is to make biodegradable implants, which will go off after some time. By the time, the cells will grow its own tissue, and it forms a functional thing. So that is the main idea of tissue engineering. So you have to choose a biodegradable polymer. You can combine 2-3 polymers and use as a single, to develop a single scaffold or something like that. So coming to the scaffolds, scaffold is a typical term which is used in tissue engineering and regenerative medicine field. So scaffolds are nothing but, when your building is constructed, that for putting concrete, there are a lot of iron rods which will be made for a support system to get a shape, right? Same way in tissue engineering also, for every organ has its own shape, its own dimension, right? So we have to get such kind of shape or dimension using this polymers or the biomaterials, which I have explained in my previous slide, will be used for developing such kind of support systems. So this support system, as I mentioned, it should be biodegradable. So it should dissociate or degrade after some time, and then whatever the cells you have seeded will form as a functional tissue. So this is the idea of developing the scaffold. The scaffold, if you define the scaffold, is like a material which provides a structural support for cell attachment and subsequent tissue development. So what I am working on is completely degradable, environmental friendly substances which will degrade after some time, because I never, would like to or would wish to work with polymers which are non-degradable, because already we have a lot of environmental issues or burden of degradability. So for the we scientists who knows that what is good and bad, we should not use such kind of biomaterials in any of our research. So coming to the specifications for tissue, TE, I have abbreviated for tissue engineering. Okay. So tissue engineering scaffold, how it should be. So porosity is the first main thing, is very important for our tissue. You can see the holes here, right, in this image. So these are the holes where it will give an interconnected pores. So as I mentioned, in our body, we have blood circulation, but that is for getting the nutrients or taking out the waste and everything, collecting the waste and everything, right? But here in the in vitro system, we don't have any dynamic system. We have a static culture, means as blood flows through and, in and out, get the oxygenated, deoxygenated blood and everything. Here in our in vitro system, there is no such system is available. We can use some bioreactors and everything, but still it is not that, it cannot mimic our body. So what we can do is we can make some pores. If it is a bulk tissue, or if it is bulky, something like this with no pores, there is no surface area also, cells kahan attach hoga, like where it will get attached. So it will be very difficult. So if we have pores in this, the cells can go inside and attach. And also one important thing is the nutrient flow will be much better, so that your tissue or whatever tissue you are aiming to develop will be able to, get into a better shape. The cells can grow and proliferate very faster. So this interconnected geometry of the pore or the porosity is very important. And pore size. You know what is the size of a cell? The size of a cell is 8 to 10 micrometers. So if you make a scaffold with a porosity of 100 micron, of course the cell is not going to stay there. It will get, pass through the scaffold. So that is not an ideal pore size which you have to develop. So you should know which cell you are going to develop. So for which particular cell you are going to seed onto a scaffolds, then only you will be able to, tweak the pore size in such a way that the cells should stay within the scaffold. And obviously it should be biodegradable, so that I have already mentioned, and mechanical performance should give sufficient mechanical strength to any tissue or any desired tissue which you are planning to develop scaffolds. And its surface properties. So the surface properties, in the sense, the cells should come and attach. So this our skin or any other part of the body is not smooth. It is fibrous. It is somewhat rough surface. So if we can mimic the surface topography of our body, it will be better for the cell attachment and proliferation. So the surface properties, we can further functionalize it. Functionalize in the sense, we can add something for the cells to come and attach. So that kind of chemical or biological functionalization we can do through reactions, which will enhance scaffold properties further. So there are different scaffold fabrication methods. So we have to choose this kind of which method we have to use also. So for example, hydrogels. You can see these are the SEM images. SEM images, and this is scanning electron microscopy image. The scanning electron microscopy, you can zoom to up to 10 micron, 10 micron resolution, and you can see the surface topography of your scaffold. That is very important. You should see whether you have pores on that, whether it is rough or not, whether it is fibrous or not, so this kind of imaging will help us to understand what is your surface topography of your scaffold. So the hydrogels, first I have mentioned that hydrogels are already, with less pores. So we cannot use it much for, regeneration purpose, but it can be used on top of skin or kind of, injectable hydrogels, which are not into a particular shape, but you can inject it. This kind of, scaffolds can be used for very soft tissues. The hydrogels don't have much mechanical properties. Next is electrospun nanofibers. So electrospinning is something what we can use for synthesizing nanofibers. So electro means some electricity or electrostatic force is used to spin nanofibers. That is electrospinning of nanofibers. So nanofibers, in the sense, the fibers can you see here, right, in this image, this will be in a nanometer range. It will be less than 1 micron. So that's why it is known as nanofibers. So the nanofibers also you can print like, not print, you can, nanofibrous mat, you can synthesize in a few millimeter thickness, and you can even make 3D structures out of it also. So these are just kind of, getting more and more popular now recently only. So other than that, you cannot see much products coming out of this kind of research. Next is gas forming salt leaching. If you see that, to enhance the porosity, we can add some salt into the polymer, and then make your polymer, make your scaffold, and then, dissolve the salt. So once salt is dissolved, wherever the salt crystals were there everywhere the pores will form. So this is known as salt leaching method. Similarly, gas forming method, some sort of forming, some sort of gas is going inside the polymer, and the bubbles will be created. Once the scaffold is made through lyophilization, that is freeze drying. Freeze drying method is like your solvent will be evaporated. So you have to dissolve your polymer in some sort of solvents. It can be water, it can be acetone, it can be ethanol, depending on the polymer solubility. You can dissolve this polymers in some sort of solvents, and then you can mix it, put it in a mold, and then you can, which requires a particular shape, dimension, whatever you require. So that can be then used, lyophilizer or a freeze dryer can be used. So freeze dryer works at minus 80 degrees Celsius. So it will freeze your components and only your solvent will evaporate. So gas forming, that is very important. Once bubbles are formed, these bubbles will be frozen like that. And once the solvent is evaporated, this gas and everything will go off. Wherever the bubbles were there, those pores will be created. So these are for enhancement of porosity further, so that the cells can sit into one. We will not get too large pores also. The porosity will be little bit controlled, so that it can, will be within the limit, like 10 to 20 microns, so that the cells can penetrate and grow and attach there, and cells will not seep through the scaffold. Then the free fabrication forms, like additive manufacturing you can use, like 3D printing. There are laser photolithography processing, and also and topography library, like you can combine different fabrication methods, like you can use 3D printing, laser lithography to develop such kind of scaffolds, so that it will be more precise in terms of dimension, or accurate shape or something. If you want to get, you can combine 2 3 of this kind of freeform fabrication techniques. So this is how the scaffolds are made. So coming back to the same tissue engineering strategy, this tissue engineering triad, we have covered cells, we have covered biomaterial based scaffolds. So now your cells are ready, your biomaterials are ready, and now what you're going to add is signaling molecules. So what are the signaling molecules? So the signaling molecules can provide some, so that we eat, or you prefer to drink Boost, you prefer to drink Horlicks. Why? The advertisement will give that you become taller, stronger, healthy, and everything, right? Similarly, our cells also should be very healthy. It should grow properly. So for that, for the proper growth of cells, we should provide some sort of Boost or Bonvita, like that. So these are the signaling molecules, we will say. The signaling molecules can be chemical cues, like it can be a growth factor, as I mentioned before. It can be small molecules, like some steroids, which for like the cells will, for to just to mimic the microenvironment of the cells. And some interfaces we can add, interface molecules, like cell to cell interaction should happen. So if the cells are growing very apart, there will not be any communication within the cells, so the proliferation won't happen. So the cells are also like a uh, social beings, like human being. If they are together, they will communicate it properly, and then only they will proliferate. So this kind of interface molecules also we can add into the scaffolds, or we can add some extracellular material cues, like if we need a synthesis of extracellular matrix. See, whatever we are seeing, the heart or kidney is extracellular matrix only, which is made up of collagen, which is made up of fibronectin or hyaluronic acid. This only we are seeing outside. We will not be able, once you are taking on organ, you will not be able to see there are cell or not. That you have to cut it, and microscopically only you will be able to see. So what you're seeing is the extracellular matrix, which is secreted by the cells. That is the complete organ. So that's why cells are very important in tissue engineering. So to enhance the extracellular matrix secretions, we can use some components like collagen itself. You can use fibronectin itself in the scaffold, or the scaffold can be made up of fibronectin or collagen or hyaluronic acid or any other polymers, or the decellularized scaffold, so that the cells will be familiar. That decellularized scaffold, the major advantage is like it is its niche, or it is the cell's home. Cells will come to know it immediately and will grow faster. And next is the physiology and metabolism to activate it. So as I mentioned, as we have blood glucose, blood serum, everything, even each single cell has that level. So that level has to be maintained very particularly, otherwise cytotoxicity can happen, cells can die. So the oxygen level, the glucose level, the serum level, the minerals level, and everything. Circadian rhythm is only for heart tissue engineering is applicable. Then immunomodulation also we can do. We can add some immune cells into that. So immune cells are the police in our body, right? So anywhere some like a thief, or we can say to us a corona virus or bacteria or something is coming inside. So our moral police inside our body will detect it immediately. What they will have start is they will start inflammation and everything. So our temperature will rise according to that. So we will get a fever. So this is how our immune cells activates. So this immune cell therapies are also available. So immune cells also can be incorporated into the scaffold if it is required for any particular therapy, like cancer therapy. Like if you require a biomaterial based cancer therapy, you can include T cells, you can include macrophages or natural killer cells or dendritic cells. So this kind of cells also can be incorporated and can be implanted near to the tumor, so that the cells will go and kill the tumor cells. And physical cues like elasticity, if we can mimic the exact elasticity, some value will be there, the stiffness or elastic value. If it is like some elasticity is like say 50 kilopascal is there. So if that can be mimicked exactly to our biomaterial, that is a physical cue which we are giving to the skin cells, which are going to seed onto the biomaterial. Similarly, the pore size, the nanopillar structure, the topography, as I mentioned before in the scaffold, is very important, because none of the tissues are smooth. There is some sort of fibrous nanopillars, some sort of roughness is there. So that roughness we can mimic using some nanopillars, nanotubes or something like that. Then again, that is a signaling molecule. These are some different sort of cues which we are giving. Then mechanical strain, like we can give compression for cartilage. Our knee joint is the largest joint which bears our whole body weight. So there in between the knee joint there are cartilages which will every time feels the compression when we are walking, running and everything. So such kind of cartilage tissue engineering requires some mechanical strain like compression or stretching. So the cells will think that they are in the same environment and they can proliferate further. So now everything we can combine together to regenerate the tissue. So the cell source, the polymers, the growth factors, this we can combine it into a scaffold, and then the regeneration can happen. So without scaffold also we can regenerate the tissue, like we can do a scaffold free method also, but the number of cells required will be very, very huge, and you will not get a proper shape out of it, and the dimensions may vary. Implantation, handling just only cells will be very difficult. So that's why scaffold method are more preferable, because it will be sturdy enough to handle, it will be sturdy enough for studying, characterization and everything. Then we will be able to regenerate the tissue. So I will be now explaining few of my studies which I have developed some tissue engineered constructs for different applications. Before coming to that, I will just introduce some other technology which can be used along with the tissue. The scaffolds are like nanostrategies, nanoparticles. So this nano size ranged particles, I think there are a lot of background noise here. So this is because Dashera is coming here in Mumbai. So it's a very big festival. So everywhere, in every nook and corner, they are bringing the Durga Devi. So that's how this dole sound you're hearing. I'm really sorry about it. So I hope it is not disturbing my presentation.
Yes,
Okay,
yes, no issues.
okay. The nano strategies also can be used, as I mentioned, there it can be a liposome, it can be a inorganic nanoparticle, polymeric nanoparticle, which can incorporate some sort of drug or growth factor for drug delivery or tissue regeneration. So why nanoparticles are because nano, I told dimension of cell is almost 10 micron. If you can prepare a nano size particle of like say 20 to 50 nanometers in range, this can go inside the cell and it can provide activity on the cell level. Like for a cancer, multiple tumor cells comprise to form a tumor, right? So if we provide nanoparticle targeted, drug delivery or something, so each and every cell will get this nanoparticle and it will exert the drug and enter cell can be tumor can be killed. That is the power of nanotechnologies, because as it comes into nano level, its surface area increases, so you have to use very little amount of drug, which is not true in case of the conventional technology where you're injecting a lot of dosages to the entire body. And if you have a tumor in your lungs, your entire body will get the drug. So that is not good, right? That's why there are a lot of side effects for the chemotherapy, why the patients are suffering a lot. This kind of side effects can be somehow we can address, or we can overcome using such kind of nanotechnology strategies. And another is immunotherapy. Already I have mentioned here in this GIF video, you can see that a T cell from our body is actually killing a cancer cell. Can you imagine? Our body has our own immune mechanism. So if we activate our immune mechanisms in such a way that it can find out the mutated cells. Cancer means it is not a normal proliferation, it is an abnormal proliferation of cell. Why it is abnormal? Because its genetic level of proliferation has some changes happened due to some external factors. That's why the cancer is developing, right? So their metabolites will be different, their cell receptors will be different, so that our immune cells initially may not be able to recognize it. That's why the cancer is getting huge. It will travel, the cells will travel through one organ to another organ, that is known as metastasis. So the cancer will spread throughout the body. So that can be prevented by uh, our immune cells itself. So we can use some tissue engineering biomaterials or the drugs which can be incorporated in the nanoparticles. If we can target these immune cells, if we can activate or if we can educate the immune cells to attack specific cells, it will go and attack those specific cells. So such kind of advanced therapies are also available. I have started working on this, but I don't have any recently we have communicated one publication on immunotherapy using T cells. So it's very interesting and promising results we have achieved for this cancer. I haven't shown in this particular presentation, but we can go through different other applications which I have developed for different developed using different biomaterials. So coming to again, the tissue engineering triad, again I am coming with the cells like mesenchymal stem cells can be isolated, it can be cultured. So first it can be isolated from a patient. So this is the main thing. The therapy is personalized, or the construct which is developed is customizable. The patient's own cells can be isolated and it can be, seeded onto a particular scaffold and it can be taken back to the patient itself wherever it is required. So it can be in the injectable form, like hydrogel I mentioned, like injectable hydrogels can be made, or it can be an implantable or surgically implantable tissue engineered construct, which can develop all these organs. But I will not say that we can develop brain. So it was there in the image, but no, I will not say we can. I will say that we cannot mimic brain. Till now, we cannot mimic actually. So coming to first, I was after my PhD, I just joined as a joined for my postdoc work. The first work which I did within collaboration with Seoul, Korea, and RMIT Australia was this nerve regeneration. So the nerves are why we are getting sensation like heat, cold, or any other like pricking, you are getting pain. This is because of nerves. So the nerves are this kind of, the single unit of nerve is known as nerve cells or neurons. So the neurons will, uh, will be like an elongated chain from each fingertip to it goes to the brain, and all the spinal cord branchings will be there. So there is a peripheral nervous system and a central nervous system. Central is in brain, peripheral is in the lower head portions, everywhere the nerves are grown. So due to some accident or something, what happens is if the network of the nerve is broken or it is damaged, you can get paralysis. This is how paralysis happens in the central nervous system due to stroke or something. You may heard that one person has got stroke, brain stroke, and half of the body got paralyzed. So this is a very common injury which happens to the peripheral, to the nervous system, which makes the entire system, entire body immobilized. So this condition is, maybe in your day-to-day life, you might have seen such kind of patients, because there are no therapy for this. We cannot regenerate nerves, but through tissue engineering, there is some hope that we can regenerate the nerves. So we, in our team, also tried to regenerate this kind of nerve tissue, which is very hard to culture. Nerve culture is very difficult compared to other culture, because the cell size is also matters, and also to provide maintain its functionality is also very difficult. So nerve and liver are the two tissues which is very tough to culture and maintain its functionality. So we have taken this challenge, whether we will be able to culture first. Yes, we could do that. And whether we will be able to regenerate it, so that we can see how we have regenerated and what type of biomaterial we have used. So first, coming to the biomaterial part. So the nerve, as I mentioned, it is an elongated structure. So it is not elongated because of the cells are connected to one each another. So that's why it is an elongated structure. So we have to mimic something that is elongated in nature. So what we have used is as a core filament is the degummed silk fibroin filament. So the silk fibroin, it is from cocoons, actually silk cocoons that can be taken out. So like silk saris and everything, that. So each silk sari is made up of single thread. So that thread, unfinished thread only we have used. So that was coated with a bio ink, we can say, which comprises of polycaprolactone and carbon nanofibers. The nerves are communicated by some sort of electrical signals, right? So this secretes kind of dopamine kind of components from one cell to another cell, and it actually electrically conducts this one, like if you're pricking your fingertip, so from there to brain it goes an electrical signal that you got a prick and you will get pain, right? So it is a matter of few seconds, that much faster the communication happens. So our material also should be conducting to pass this sort of electrical signals, which is through the cells which are getting used for communication of one between communication between the cells. So the degummed silk fibroin which we have used was coated with a conducting material. So why PCL is like, PCL is also a FDA approved, Food and Drug Administration approved biodegradable polymer. Silk fibroin is also Food and Drug Administration approved biodegradable polymer. So this combined, and we use this thread, and then we have used to make textile structure like braids, like how you braid your hairs. That is one structure we use. This is very micron level. Okay, don't think that it is very big braid or something. So this is the scanning electron microscope. You can see this entire thing under a microscope like this. This is a knitted structure, knitted in your sweater and everything. You see this knitted structure, right? Similarly, we also knitted the scaffold, and here this is the knitted structure, and there is an outer covering, because if we implant this to the damaged nerve gap, So what happens is other cells also can come and proliferate. So we want to prevent that. So only the nerves to nerve end has to be bridged together. So what we have did is we have made a one layer film which is non-porous, so that the cells should not go inside, but it is a hydrogel. So that kind of film we have made and used as a covering of this kind of nerve conduit. So this is called as conduits. Conduits in the sense longer conducting wire or something, connecting wires and all, you can say that. So this kind of conduits we have made. So this is a larger picture of the knit. We have rolled those fibers. You can see the single fiber. This is 200 micron size. So the single fibers, you can imagine what is the size of it. These are in a micro level size. So the braids we have made, and this is the complete conduit which has inner core and outer film. So this is a video which I would like to play. In my pointer option, the arrow is not coming. So we have used a New Zealand white rabbit for implantation of the nerve conduit, and we have implanted it. And so after 7 days of surgery, you can see the right side, I think. Yeah, right side of leg we have used. That leg, it is not able to move. But after 30 days, see, it can move its leg. The same rabbit we have used after 30 days. So this end of 30 days, we have surgically removed it and studied its potential extensively. So we could see that the functional nerve regeneration was happened. That means we can prevent now paralysis. So this kind of, if it can come into the market, it can give a greater potential for regeneration of damaged nerves. Even we have 3D printed the nerve conduits. So 3D printing method, I think you already know how it is. So what we have made is like the bio ink was made up of PCL, and we have given an extra growth factor or a signaling molecule, that is dopamine, and also the carbon nanofibers. That's why this ink is black in color. So the carbon nanofibers, we optimize all these concentration and everything and loaded onto the 3D printer, and we have made lines of it. So this is the 3D printed film. So here you can see that the 3D printed line is shown here, and the cells proliferation, this is animated just to show that how the lines are there. Can you see here in this image? This is a confocal image where the cells can be stained. We can use different colors to stain the cells. Like this is a fluorescent stain which we have used. The blue one is DAPI, which can stain the nucleus of the cell. Here you can see in the animated image there is a nucleus and there is a other extracellular matrix and the phenotype of the cell you can see. So the phalloidin, or the filaments of the cells was stained using phalloidin, that is pink in color. So when it is merged, you can see the blue and pink color. So wherever the lines were there, which we have printed the lines there, we can see that the cells are proliferating on top of the line. It is not going outside. Why? Because it has a signaling molecule dopamine there, but other side it is not there. But your control, you can see it is just growing here and there. So this is a chemokine, as I mentioned before, and also it is a signaling molecule which helps the cells to come together and grow in some direction wherever we draw the line. So next work we did is hernia mesh. Hernia, I think, is a very common disorder. What is hernia is like? So we know that our stomach, below that there is it's a covering is skin everywhere, yeah, obviously. Below that there is fat pad, and then comes the intestine, right? So what happens in hernia condition is like our intestine, due to other pressure or some kind of obesity or some other condition, the pressure get increases in the stomach area. So what happens is this intestine can come out like this. So once the intestine come out like this, what happens, it will start paining, it can start the infection, it can even rupture the skin. So there will a lot of other complications, and as it is in intestine, our digestion also will not be proper. So what doctors do is, so there it is like they will push back the intestine like this. Here you can see a rod, right? It's not a rod actually, it is a film, it's a mesh. So this is a hernia mesh. So they push back the intestine back to its normal position. They anyway, it happens surgically only. They will keep a mesh on top of the intestine. They will suture it to the fat pad, so that the, even though there is a lot of pressure also, this kind of fibrous structure will not allow the intestine to come back. But some other area it can happen. We cannot put mesh throughout the stomach. So wherever the problem is there, this can be a solution. So this is a mesh like structure which we also developed. So this is the mesh which we have developed using polycaprolactone, and we have coated it with nano diamonds. So we have seen diamonds in very big structure, crystal structure, but here we have used nano diamonds. This is why, because it can prevent the biofilm formation. That means, so as it is just below the skin, anytime inflammation can happen, because during the wound healing, so this kind of infections we can prevent when we have nanodiamond coating on top of this kind of meshes. So we have developed this kind of hernia mesh, and we have tested, this was kind of an industrial project which we have coated and given as a product to them. And now maybe in near future, this can come as a product. So this work was in collaboration with RMIT Australia, this nano diamond hernia mesh. So coming to the another work which I have done is like 3D printing of nanocellulose. So cellulose is toughest, we can say, toughest natural biomaterial. So this is very difficult to even print or something like that. So what we have used, we didn't use the conventional available cellulose powder and then make a gel or something like that. We have synthesized cellulose through microbial reaction or a fermentation product. Have you heard of kombucha tea? Have you heard? Kombucha tea, you can search in Google. It's very healthy drink. You can also have it. It has a lot of antioxidants and everything. So this kombucha tea is is produced through this kind of fermentation, microbial fermentation, that is through SCOBY. Is the SCOBY you can see, right? This is the seed material for making kombucha. So the seed material is symbiotic association of bacterium and yeast. So this two, when comes together, it will start making some type of film. So this film can be used as a leather also. This is bio leather, animal cruelty free leather also, because it is such a tough material. This you cannot use it for any particular application, because there is no flexibility to use it into different forms or shapes or like that, right? So what we have did is, this leather we have hydrolyzed using acids, and it was hydrolyzed in such a way that it forms a gel and we can 3D print it. So this is how we have 3D printed. This is the kombucha culture. You can see this video where the 3D printing happens. So we have printed a simple lattice structure just to see the feasibility of 3D printing out of it. And further, the cellulose, anyway, it is used widely for wound healing and everything. So this can be also used for wound healing applications. Oh, it's 3:43. Sorry, it's too long, I think. Anyway, a few more slides. I will just conclude. Is it okay? Like I can explain a few more slides if there is time?
Yes, you can take up to 5 minutes.
5 minutes, okay. So this is another research which was published. So we have used cartilage. We have developed a construct for cartilage regeneration. So as I mentioned, human knee joint is the largest joint, right? So here, to bear the strength of the entire body, so if it is like 60 kg is fine. If it goes beyond 80 or 100 kg, the body is not meant to, or organelles are not meant to withstand that much weight. So what happens is, this cartilage, you can see the C-shaped or cashew shaped, this cartilage will get teared and it will go off, and what happens is the bone to bone friction will happen, then that that arthritis will come, osteoarthritis. It's a very common disorder nowadays because of it's a lifestyle disease. So because our lifestyle is not proper, we are not exercising, we are not eating, our eating pattern is not good, that's why this kind of disease happen. And also due to accidents and all this can also hereditary and also can happen, but mainly we can classify it as a lifestyle disease. So this is the scaffold which we have developed for using silk fibroin. So this is the cocoon. Silk fibroid was isolated, and this was processed, and we have made this kind of scaffolds, and we have studied it in rabbits actually, and we could see that you can see the blood vessel formation and everything inside the scaffold. There are multiple components which was added into this scaffold to make it appropriate so that it can withstand the weight as well as it can regenerate and proliferate the cells. So coming to the one more study is the recent study which I did in IIT Bombay. It's like development of a dermal patch which can give multifunctional aspect where it can give antibacterial, antioxidant, anti-inflammatory, as well as it can provide, it can have some chemoattractant where the fibroblasts can come and regenerate the wound area. So this is the dermal patch. Two forms I have developed, that is film-based and one is nanofibrous patch. So this violet patch, which is made up of different polymers depending upon the the functionality which I require. So that's why I have chosen different polymers. So that was attained using this combination of polymers and bilayer structure. And then this is the SEM image of this polymers. And we did the wound healing studies in Sprague Dowley rat. So we have used a positive control where you can see the wound has to be healed a little bit more. But in case of this scarless, wound healing was achieved using our technology. So this is also we are going to we have already patented, and this is coming to the market soon. And as I mentioned, anything we can any tissue or any components can be tissue engineered, but why it is not coming to the market, why it is not readily available for now. Also the cancer patients has to go through chemotherapy, there are no nanoparticles or something fancy things are given for whatever I have mentioned is not given as a treatment. Why? Because of set regulations. There are a lot of regulations. Food and Drug Administration has a strict regulations that we have to go through, and a lot of protocols to get something from the bench or the lab to the bedside. So it is very difficult. It is a tedious process because every institute will not have such kind of facility to do all these testings and to compile into a document and to submit to FDA and get approval. No one will wait that much. So the one of the in vitro, these are the enlisted in vitro analysis we have to do if we are using a biomaterial. So there has if we are using fibroblast, if we are using endothelial cells, if you're using blood cells, if you're using immune cells, if you're using some kind of targeted tissues we are doing, or according to different tests, there are different tests we have to do. And in vivo analysis also, there are we have to choose a proper animal model. And for getting clearance for animal model is also a tedious task, because we can do in India, the CPCSCA, it's the control of animals ethics for experimentation, that who gives us the permission to do the testing or experimenting in animals. Most of experiments will happen in rabbit, hamster, guinea pig, or rabbits. Higher animals like monkey and dogs are really rare, and it's very difficult to get approval also. So lot of approval processing we have to go. So this is the overview of the risk management and how the product is coming into the market. So we have to do in vitro analysis, then preclinical evaluation, that is the in vivo study in animals, then clinical evaluation. So once the in vivo analysis is done, the results will be checked. It has to be repeated multiple times. And then if we are getting a reproducible result, then we can do the clinical evaluation, that is in the human beings. So the in human beings to getting, no one will get ready to do some kind of this kind of experiments in, whether you will be ready for doing some kind of experiments, please come to me. So it will be very difficult to get human subjects to do some kind of clinical evaluation. So we have to target patients. For that, a lot of funding is required. We have to fund that single patient for their travel, their treatment, everything. We have to take care. So the fund crunches are also there. Then only once the human clinical evaluation is done, then only we can get it into market. If at all the quality, the safety and efficiency we can guarantee 100%, then only it will come to the bench to bedside. So that's all about the tissue engineering in biomedical field. Thank you so much. I'm happy to take any questions if you have.
Thank you, ma'am. There is a question in the chat box.
Yes, please.
I'm reading the question. Are the prepared cells are used as filament in 3D printing?
So there are different types of 3D printers. So one type is used filaments, that is extrusion printers, which is high temperature based, like 80 degrees, 90 degrees, 120 degrees and all. So here we cannot withstand then how a single cell will withstand. So there are sophisticated printer like 3D bio printers for printing cells. So the filaments are not used. Maybe you can 3D print the filaments based extrusion printed scaffold, and later you can seed the cells, but you cannot print it along with the filaments if it is of high temperature 3D printing.
Okay. In your experiment, you have used some rabbits. What kind of efforts or effects does the rabbit gone through?
So
effects means some temperature rise or something related to its physical.
Okay, I got the point. Yeah. So the question is basically, so for any experimentation, the rabbits or any animals which we have to use will be in a proper condition, will be maintained. So these are known as animal houses for experimentation. So CPCSCA in India, so this they will give approval for animal house if only they are maintaining the animals properly. The body temperature, their feed, their sterility, their water, they should give a sterile water, sterile oxygen, everything. If it is there, if they have a sophisticated facility, then only they will approve for giving a permission to do or permission to even procure the animals and keep there. So we don't have to, so we will go and do only experiments in such kind of animal houses. So the other parts of whatever other than surgical pain and everything, other than that, it's feed, temperature, everything is taken care.
Hi, good evening, ma'am.
Hi.
It has been a very nice talk. Actually, I don't know much about 3D printing, ma'am. Whether we will be, I have a question, ma'am, whether we will be able to adjust the porous size using this 3D printing, whether it's possible to design as a scaffold with varying porosity sizes, whether it is able to adjust the porosity using 3D printing.
So the 3D printing, actually, as you told, you don't know about. So the
yeah,
3D
ma'am. Yes, ma'am. Yes, ma'am. I'm new to 3D printing.
Yeah. So the 3D printing is basically a computer programmed device, sophisticated equipment. So where you can design your pore size, where you can design your filament structure, scaffold structure, everything. So if you need a 10 micron pore size, you can design your scaffold in such a way that the 10 micron pore size is achieved with some sort of thickness and some kind of shape. So it is very precise in terms of the precision is very high in terms of whatever shape you have to achieve, whatever the size you have to achieve. So it is completely computer programmed equipment. So you don't have to worry about whether you will be able to tweak the porosity or something. It is possible. Yes.
Okay, ma'am. Okay, ma'am. Thank you.
Thank you.
Okay, I think we shall conclude this session. So with this, I thank you for the wonderful presentation. Now it's time for the formal vote of thanks. First of all, I would like to thank the management team of the Madurai M.D.T.Hindu College for their support and their continuous encouragement. Next, I thank Dr. A. Subramanian, Principal, President IAC, Dr. K. Balasubramanian, Coordinator IQAC, Vice President IAC, Dr. P. Velmani, convener IIC, and to the other organizing committee members, Dr. V.C. Sankar, Dr. A. Sivagurunathan, Dr. A. N. Seepalasmi, Dr. A. S. Ganga, Dr. K. Viswasarojini Devi, Dr. C. Anita, Dr. S. Logambal for their backing support. Next, I would like to thank Mr. R. Subhash Chandra Bose, Assistant Professor of Physics, Management Teaching Staff for the technical arrangement of this event. At an outset, on behalf of all of us and the Madurai M.D.T.Hindu College, I thank the resource person, Dr. M. Mamata Pillai, very young, active, dynamic researcher, to accept immediately the invitation for this webinar, owing to her physical conditions and her busy schedules, and for the wonderful presentation. Thank you. And I thank all the participants for your cooperation during the presentation of this online webinar. Thank you. Thank you all.
Thank you so much for having me. Thank you for the organizers once again. Wish you all the best.
Thank
Bye.
you. Thank you.